Assalamu alaikum, dear students. In this lecture, we will talk about the randomized control trial. We will discuss about the advantages, disadvantages of the trial, the types of the trial, as well as we will also talk about the different bias and their examples in the during the study, before the study, and after the completion of study. So, what are the subtopic or the objective of today's lecture? Initially, we will talk about the principle of RCT. That what are the key principle of the randomized controlled trial. We will also discuss about what is the RCT, then the types of RCT, the biases, and as well as their types and advantages and limitation of the RCT. सो अगर हम बात कर ले की प्रिंसिपल ऑफ आरसीटी की, तो उससे पहले थोड़ा सा हम आरसीटी की बात कर ले कि ये आरसीटी इसको क्यों कहते हैं. सो रेंडमाइज्ड कंट्रोल ट्रायल, ये तीन वर्ड्स है. ट्रायल मीन इट इज एन एक्सपेरिमेंट. कंट्रोल्ड का मतलब ये है कि इसमें हम एक कंट्रोल ग्रुप ले लेते हैं. कंट्रोल ग्रुप से मुराद ये है कि ये इससे हम कंपैरिजन करेंगे एक्सपेरिमेंटल ग्रुप का. और अमूमन ये ऐसी ट्रीटमेंट पर मुश्तमिल होता है जो स्टैंडर्ड होता है, जो एविडेंस हाई क्वालिटी एविडेंस जो है, टॉप क्वालिटी एविडेंस जो है, इस जो स्ट्रेंथ उसकी ज़्यादा हो, सिस्टमेटिक रिवीजन मेटा एनालिसिस वो उसको सपोर्ट करता हो, वो ऑलरेडी जो है इस एविडेंस मौजूद होती है कि ये इसकी इफेक्टिवनेस जो है ये है, और ये एक पॉजिटिव इफेक्ट वाली ट्रीटमेंट है. या जो है एक प्लेसिबो भी हो सकती है विच एक्चुअली लुक लाइक अ ट्रीटमेंट बट इट्स नॉट एक्चुअल ट्रीटमेंट, जस्ट लुक लाइक अ ट्रीटमेंट. तो लेकिन हम उससे कंपैरिजन करते हैं तो ऐसी एक्सपेरिमेंट जिसमें कंट्रोल्ड होता है, दिस इज नॉन एज द कंट्रोल ट्रायल. इससे पहले जब हम स्टडी क्लासिफिकेशन डिजाइन क्लासिफिकेशन डिस्कस कर रहे थे, तो वहाँ पे हमने डिस्कस किया था कि जितनी भी एक्सपेरिमेंटल और इंटरवेंशन स्टडीज हैं, they can be divided into two types: the controlled trials and the uncontrolled trials. Uncontrolled trials, which जिसको हम प्री एंड पोस्ट स्टडी कह सकते हैं, जिसमें एक ग्रुप हो सकता है, सिंगल ग्रुप से डिजाइन होता है, जिसमें हम सिर्फ और सिर्फ उससे ट्रीटमेंट से पहले ट्रीटमेंट के बाद देख लेते हैं, यानी देयर इज नो सच कंट्रोल. कंट्रोल्ड ट्रायल दे कैन बी देन डिवाइडेड इनटू टू टाइप्स. हमने डिस्कस किया था कि randomized control trials and non-randomized control trials. सो नॉन रेंडमाइज कंट्रोल ट्रायल पर डिटेल हमारी बात हो चुकी थी, कि उसमें इम्प्रॉपर रेंडमाइजेशन वाली भी आती है, क्रॉस एक्सपेरिमेंट वाली भी आती है, विदाउट रेंडमाइजेशन भी आती है, और एग्जांपल भी हमने डिस्कस किया था कि प्रॉपर इम्प्रॉपर रेंडमाइजेशन से मुराद क्या है, क्रॉस एक्सपेरिमेंट स्टडी से मुराद क्या है. आज हम इस लेक्चर में हम रेंडमाइज्ड कंट्रोल्ड ट्रायल पर बात करेंगे कि इसकी टाइप्स क्या है, इसकी लिमिटेशन क्या है, इसके एडवांटेजेस एंड डिसएडवांटेजेस क्या है, और ड्यूरिंग द कंडक्शन ऑफ दिस आरसीटीज क्या क्या बायसेस जो है ये हो सकती है. सो थोड़ा बहुत हम आरसीटी की तारीख पर कि भाई ये कैसी इसकी इवॉल्वमेंट होगी. तो बेसिकली आरसीटीज जो हैं दे वर इवॉल्व फ्रॉम द फार्माकोलॉजिकल एंड मेडिकल प्रोफेशंस. स्पेसिफिकली ड्रग एक्सपेरिमेंट से इसकी शुरुआत हुई थी, और इस डिजाइन में, इस एक्सपेरिमेंट स्टडी में ट्रीटमेंट जो है ये रैंडमली एलोकेट किया जाता है, एंड मिनिमम देयर विल बी टू ग्रुप्स. ज्यादा ग्रुप भी हो सकते हैं, थ्री भी हो सकते हैं, फोर भी हो सकते हैं. एंड इफ सपोज इफ देयर आर ओनली टू ग्रुप्स, सो दैट इज कॉल्ड एज अ टू आर्म स्टडी. एंड इफ देयर आर थ्री ग्रुप्स, different groups receiving three different treatments, so they will be called as the three arm study or the three arm trial. सो यहाँ पे ट्रीटमेंट जो है ये मुख्तलिफ ग्रुपों में ये रैंडम एलोकेट किया जाता है. That is not upon the choice of the patient or upon the choice of the researcher. So that is randomly allocation of the treatment to the different groups. Similarly, the patient, the subjects, they are also allocated to the different groups through the randomization. We have discussed in the previous lecture that what are the three common methods of randomization, that how we can do the randomization, and in the study where randomization is present. So different treatment groups should be statistically equivalent. और ये भी हम डिस्कस किया था और ये थोड़ा सा मैं रिवाइज कर लूँ कि स्टैटिस्टिकली इक्विवेलेंट से मुराद ये है कि दे शुड बी इक्विवेलेंट, दे शुड बी इक्वल एट द बेस लाइन। जब उनकी ट्रीटमेंट अभी तक शुरू नहीं है, सो इट मीन देयर शुड बी नो सिग्निफिकेंट डिफरेंस बिटवीन द ग्रुप्स, बिकॉज वहां पर अगर ग्रुप डिफरेंस होगा तो फिर ऑब्वियसली हम रिजल्ट कंपैरिजन में बायसनेस हो सकती है। सो देयर शुड बी बेसलाइन सिमिलरिटी before the treatment when they are going to start the treatment, and aim to establish even efficacy of a treatment and to detect the side effect associated with treatment. So that is very good. Much of the studies that do not report the side effect, but it doesn't mean that they are just hiding the side effects. Sometimes there are no side effects, that's why there is no reporting. But it is too good if there are side effects or there are no side effects. In both cases, it should be mentioned about them. If there were no side effects reported, so it should be reported in the literature, in the study, in the manuscript, that there are no side effects were reported in the subject. And if there are some side effects, it should be then must reported in the manuscript in the results of that study. Another characteristic of RCT is it is there is not only just we discuss the random treatment, but there is also the random allocation of participant to the treatment or control group. Treatment group mean experimental group, and control group which we have discussed earlier. Both groups are followed by a specified period. Groups are analyzed in terms of outcome defined at the onset, and there is a focus on the control of bias. Now it is the responsibility of the researchers not only to identify the bias, बल्कि उसको ये कोशिश भी करनी चाहिए कि उसको इसको कंट्रोल कर सके, इसको कम से कम कर सके, तब जाके हमारे रिजल्ट जो है थोड़ी सी अनबायस्ड आएंगे। अब मेजर सोर्सेस ऑफ बायस हो सकती है, इसके अगर हम क्लासिफिकेशन कर ले तो कुछ biases जो है they can be just before the start of the study. They can be called as the pre-trial bias, which are related to the planning of the trial. Some biases they occur during the trial when already there is implementation of the trial, when there is the treatment implementation protocol. These trials they are also known as the bias during the trial or in trial bias. While some biases they can be after the completion of the study. They can be occurring during the data analysis or they can be occurred during the publications. And these are known as the post-trial bias or also known as the bias after the trial. Now, if we talk about the bias which occurred during the planning of the trial are collectively known as the pre-trial bias. They can be of different bias just like the plot study design, the selection wise or the channeling wise. अगर हम अभी प्लॉट डिजाइन की बात कर ले कि what we mean by the plot study design, so actually a plot study design there refers to the that research that is improperly structured, and this improper structuring of that study they will lead to the unreliable or unvalid result. और ये जो क्लास होते हैं इस ड्यूरिंग द स्ट्रक्चरिंग ड्यूरिंग दिस प्लानिंग ये मुख्तलिफ रीजन से हो सकते हैं, मतलब दे कैन अराइज फ्रॉम द methodological issues, they can be the failure to control confounding variable, but identifying and addressing these flaws they are very critical for the producing the credible results, credible findings. अगर हम बात कर ले कि कॉमन कॉमन फ्लॉज इन स्टडी डिजाइन क्या क्या हो सकते हैं, सो इफ वी आर conducting the randomized control trial and there is lack of randomization, so this is one of the common and the most critical bias in this planning during the planning of this trial, because without random assignment groups may differ in ways unrelated to the intervention leading to the confounding. And for example, a clinical trial that assigned treatment based on the patient preference, leading to the uneven distribution of the disease severity. दूसरे एग्जांपल या हम कह सकते हैं कि कॉमन फ्लॉज इन द स्टडी डिजाइन जो हो सकती हैं, दैट इज द inadequate sample size. अगर स्मॉल सैंपल साइज जो है ये हम ले लेते हैं, so actually a small sample size, it reduce the statistical power and it also increase the likelihood of the type 1 error or the type 2 error. Type 1, type 2 error already हम discuss कर चुके हैं in the previous lecture. Type 1 error, which is also known as the false positive, and type 2 error, which is also known as the false negative. This type 1 error is also known as the alpha error, and the type 2 is known as the beta error. अब मिसाल के तौर एक स्टडी जिसमें हमने 10% पार्टिसिपेंट सिर्फ लिए हैं. So these 10 participants may not detect a significant effect of an intervention. That's why if we want to a significant effectiveness of a treatment in a trial, there should be enough sample size in both the groups. The next is the selection bias. Selection bias, that simple mean that the participant they are not representative of target population. We have some problem in the selection in the recruitment of the participant. We have not recruited them properly according to the inclusion and exclusion criteria. So there may be some problem in the screening, and due to them, the resultant bias is known as the selection bias. For example, a study on exercise effect only include athletes, excluding the sedentary individual. Now, यहाँ पर the athletes they are must their body is built up, their body is adapted to the exercise, and they are regularly doing the physical activity. So that's why the effect of the exercise on them will be much more different than those who are not doing any physical activity in the routine in the sedentary individuals. Uh, इस द सिलेक्शन प्राइस और पार्ट थ्री डिजाइन के अलावा जो एक और हमारे यहाँ पे ड्यूरिंग द ट्रायल हो सकती है, जो यहाँ पे मेंशन नहीं है, वो यही है कि when we have the lack of control group or when there the lack of identification or any procedure for the identification and controlling the confounding variable. Because if there is failure to control for the variable that influence both the exposure and both outcome, which influence both the dependent and independent outcome, so obviously it will lead towards the biased result. For example, a study linking coffee consumption to heart disease overlooks smoking habits in the coffee drinkers, and that's why that may have resulted in biasness in the result and finding. Similarly, if during the construction, during the planning, if you don't have a control group, so without a comparison group or control group, it's impossible to determine if changes are due to the intervention or it is due to some other factor. For example, observing weight loss in participant on a diet without comparing them to the non-dieters, that will be not a wise decision, not a wise design. So there are other common reasons for the uh, plot study design, like the measurement bias, like mean we have inappropriate study design, like choosing a design that does not make the research question. There may be the observer or the researcher bias. Similarly, if sometimes we don't have the blending, though it is not mandatory that every study should have the blending, but if there is blending in the study, so it has a very positive effect, specifically the patient and the assessor. And more specifically, the assessor blending is very necessary because if the participant or the assessor known to the group assignment, this can influence the behavior or the outcome assessment. And that's why the assessor they should be kept as blind. And if they are not blind, so there may be a bias, there may be during the pre-trial bias because we don't have considered them in the design. We don't have considered blending during the trial planning. Some other bias like the channeling bias. Now, what we mean by the channeling bias? The channeling bias that may refer. Okay. सो चैनलिंग बाइस से मुराद ये है कि हम किसी ऐसे जो पार्टिसिपेंट है उसको हम चैनलाइज करें अकॉर्डिंग टू देयर एज अकॉर्डिंग on the basis of their age, on the basis of their gender, on the basis of their intervention, and regardless to what group they are assigned. It means if someone is randomized to a group A, but after that we, on the basis of some specific characteristic, we channelize them to the treatment B. So that is actually a bias, and it means we are going against the randomization. हमने randomization की है और उसने कंट्रोल ग्रुप में है और फिर इसको हम एक्सपेरिमेंट बिकॉज ऑफ द सिवियरिटी ऑफ द डिजीज बिकॉज ऑफ द जेंडर बिकॉज ऑफ सम एनी अदर फैक्टर we just shift them into the next group so that we call is the channeling bias. So channeling bias sometimes it we can say that may have a critical role in creating the bias in the result because here there may be disturbance the number of participants in both the control and the experimental group. Some biases they can be occur during the implementation of the trial mean already the um, trial has been started and during the trial वहाँ पे कुछ गलतियाँ कुछ bias हमसे हो सकती हैं. And among them, there is the interviewer bias, the chronology bias, the recall bias, the transfer bias, the misclassification of the exposure of outcome, or the performance bias. So we are going to discuss each one of them. सबसे पहले जो है interviewer bias. So interviewer bias, these types of biasness they occur when the interviewer action, attitude, or expectation they have some influence how the participant respond during a study, and this influence they will obviously lead to the distorted and unreliable data. We can also elaborate this interview bias that it is a type of measurement bias that can commonly arise in surveys, interviews, or any research requiring the interaction between the researcher and participant. Now, what are the main causes of this interviewer bias? Interviewer bias से मुराद ये है कि जो रिसर्चर क्वेश्चंस करता है टू पार्टिसिपेंट से उस क्वेश्चन में वो ऐसी कुछ क्वेश्चन को चेंज कर लेता है अपने फेशियल एक्सप्रेशन से या कोई लीडिंग क्वेश्चन से दे रहे थे जिससे पार्टिसिपेंट की जो अपनी ओपिनियन होती है वो थोड़ी सी चेंज हो जाती है उसकी जो अपनी परसेप्शंस होती हैं वो थोड़ी सी चेंज हो जाती हैं सो इफ वी लिस्ट दैट व्हाट आर द काजेस ऑफ द इंटरव्यूज बायस तो सबसे कॉमनली जो अक्सर स्पेसिफिकली इंटरव्यू और क्वालिटेटिव स्टडी में मोस्ट कॉमनली होता है दैट इज द लीडिंग क्वेश्चन लीडिंग क्वेश्चन मीन कि आप पेशेंट से जब आप इंटरव्यू लेते हैं और उसको आप एक हिंट दे देते हैं कि आप इससे एग्री करते हैं कि ये पॉलिसी जो है ये बहुत फायदेमंद है तो सही इस तरह है आप इस तरह है ना आप एग्री है तो आप उसको पहले से जो है क्वेश्चन लीडिंग क्वेश्चन दे देते हैं कि आप यू एग्री दिस पॉलिसी इज बेनिफिशियल राइट तो आप उसको आंसर भी दे देते हैं और प्लस उससे क्वेश्चन भी करते हैं तो वो कहेंगे यस I understand it's very beneficial. So that is actually interviewer bias just because of that leading question. Somehow, in other case, maybe the non-verbal cues. जरूरी नहीं है कि वहाँ पे वो उसको leading question और verbal cues दें. मिसाल के तौर आप एक रिस्पोंडेंट एक ओपिनियन दे रहे हैं जो एक क्वेश्चन की सूरत में कर रहा है एंड द रिसर्चर द इंटरव्यूअर उसको नॉडिंग कर लेता है या एक स्माइल दे देता है और उसको पता चल जाता है कि यस आई एम ऑन द राइट वे या आप सही बोल रहे हैं तो फिर वो आगे जाके उसको कंटिन्यू रखते हैं और उस वहाँ पे वो थोड़ा सा नो में सर हिला देता है तो वो अपना रिस्पॉन्स चेंज करता है कि उसको पता चल जाता है कि मैं सही नहीं बोल रहा हूँ तो एक्चुअली ये भी जो है इंटरव्यूज बायस में आता है इसी तरह सेलेक्टिव रिकॉर्डिंग की जब आप उनसे इंटरव्यू लेते हैं और उनसे क्वेश्चन आंसर सेशन करते हैं तो उसको हम रिकॉर्ड करते हैं तो आप कुछ जगह जो है कुछ आंसर क्वेश्चन आंसर सेशन को रिकॉर्ड करते हैं और कुछ को आप रिकॉर्ड नहीं करते, इंटेंशनली आप रिकॉर्ड नहीं करते. So basically, this type of they can also lead to the interviewer bias. इसके अलावा जो है, पर मिसाल के तौर जिसको हम हमारे कुछ एक्सपेक्टेशंस होती हैं, और वो एक्सपेक्टेशन जो है हम पार्टिसिपेंट से चाहते हैं, और हम कोशिश करते हैं कि इसी तरह क्वेश्चन करें, और इसी तरह क्वेश्चन भी हम उनको समझाने की कोशिश करते हैं. लाइक द इंटरव्यूअर्स assumption अबाउट द पार्टिसिपेंट बेस्ड ऑन द डेमोग्राफिक्स. हम देखते हैं कि उसकी एज क्या है, उसके जेंडर क्या है, उसकी इथनिसिटी क्या है, और फिर उसी की रिलेटेड हम उससे एक क्वेश्चन कर देते हैं. यानी यानी हम सॉरी यानी हम उसकी डेमोग्राफिक वेरिएबल के बेस पर उनसे कुछ डिफरेंट क्वेश्चन, एक से करते हैं, दूसरे से अलग क्वेश्चन करते हैं. So that's just because we are expecting some answers from the older people, some other answers from the young people, and the base of ethnicity. So just because we want answer according to our expectations, so we change the answers just on the basis of the different demographic variables. इसी तरह एक और कॉमन काज हो जाती है जो ड्यूरिंग इंटरव्यू बायस का बायस बन सकती है. दिस इज द इनकंसिस्टेंट क्वेश्चन. कि सपोज आप 5 बंदों से इंटरव्यू ले रहे हैं, एक बंदे से आपने इस तरह की क्वेश्चन की, दूसरे से आपने और किस्म की, तीसरे से भी आपने सेम क्वेश्चन की, लेकिन इसमें से एक दो क्वेश्चन आपने लीडिंग क्वेश्चन ऐड किए, आपने नए क्वेश्चन ऐड किए. तो ये जो चेंजिंग द प्रेजिंग टून ऑफ क्वेश्चन अक्रॉस द पार्टिसिपेंट, दे में आल्सो लीड टुवर्ड द इंटरव्यूअर बायस. For example, probing for more details only when answers align with the interviewer expectation. So that may lead towards the interviewer bias. So बहुत सारी इसकी examples आते हैं, but we will just go towards the chronology bias. What is the chronology bias? Chronology bias, it occurs when the timing of the study or the intervention that influence its result and it lead to the biased conclusion. It is particularly relevant in research where temporal factors such as advancement in treatment or change in the healthcare practices that affect the outcome. There are a number of causes which can lead towards the chronology bias, like the historical context, which mean the difference in the medical practices, technology, or the social norms that no, they are conducted over time, and also they will have influence study outcome. For example, you are going to compare the patient of cancer from the studies which were conducted in back in 1990s and those studies which have been conducted into 2020. So there is a big difference between the 1990s and the 1920s. So there's difference that will lead to that chronology bias, and इसको हम कहते हैं the cause of the chronology bias is the historical context. Another cause of the chronology bias that may be the sequential enrollment, which means in non-randomized studies, participants enroll at different times. They might experience different conditions. For example, a patient receiving treatment during a pandemic may have worse outcomes due to the resource limitation. There may be another cause of the this chronology bias like order of data collection. For example, when data is collected or analyzed in phases, an early result they might differ from the later one due to the changes in the protocol or some other external factor. So all these causes, either it is the sequential enrollment, either it is the order of data collection, or the other which we discussed in the start, the historical context, they all may lead towards the chronology bias. The third one is the recall bias. Recall bias means when you are asking the participant about their past and they are not remembering the complete statement, the complete situation, and they are partially remembering and partially they have forgotten. So जब वो आपको बयान दे देते हैं तो उसमें से कुछ चीजें जो भूल जाती है, या तो वो मिस कर देते हैं या उन नई चीजों से, जो फाल्स चीजों से उसको रिप्लेस कर देते हैं. तो चूंकि यहाँ पे बहुत या तो फेब्रिकेशन करता है या फॉल्सिफिकेशन करता है, यानी या तो डेटा कंस्ट्रक्ट कर देता है यहाँ पे, अपनी तरफ से कुछ बोल लेता है जो एक्चुअली वो रियलिटी में ऐसा कुछ नहीं होता, या जो है जो कुछ हुआ होता है उसको थोड़ा सा वो चेंज करके बताता है जस्ट बिकॉज यू आर नॉट रिमेम्बरिंग प्रॉपरली. So that type of biases that is known as the recall bias. The next is the transfer bias. Now, what we mean by the transfer bias? Transfer bias, they usually occur when there is a systemic difference in how the participant they are moved, they are transferred between the groups, between different hospitals, or that may be in the different stages of study as well. And all these will lead to a biased result. This type of bias, the transfer bias, is particularly relevant in observation studies, the cohort studies, but they may also be very critical and important in the clinical trial as well, where patient follow-up or transition impact the study outcome. What are the different causes of the transfer bias? So the one of the common is this unequal lost follow-up. Uh, Participants who drop out are transferred to another group may differ significantly from those who remain. For example, in a study comparing the treatment for stroke patients who experience complications may be more likely to leave the study, which will obviously skewing the result. There another reason for the transfer bias there may be the systemic difference in transfer. Patient transfer to different settings, for example, there may be the rehabilitation center, there may be the hospital, uh, and these patients may receiving the different level of care. For example, there is one protocol, one sort plan of the treatment in one hospital. There is another protocol in another hospital. So when they are shifted, when they are exchanged between these hospitals, when they are transferred between these hospitals, so obviously they will receive the different type of care, which will be not similar to among the group members, among the participants of that trial. So that will lead to the biases, and that such biases is known as the that transfer bias. Similarly, when there is during the follow-up or data collection, they may differ between the groups after transfer, leading to incomplete or inconsistent data, and that will lead to also called as the transfer bias. The next one is the misclassification of the exposure and outcome. In this sort of bias, usually the one which is exposure is considered as the outcome, and the one which is outcome is considered as exposure. And this wrong consideration that is actually known as the misclassification of exposure and outcome. ये जो bias है ये तब होती है mostly when we are unclear about the cause and effects relationship, जो temporal relationship है वो clear जब नहीं होता. ज़्यादातर ये studies जो हैं ये उसमें जो केस कंट्रोल स्टडी है उसमें बहुत ज्यादा कॉमन होती है, क्योंकि वहां पे ऑब्वियसली हम रेट्रोस्पेक्टिव होते हैं, तो वहां पे ये क्लियर काज एंड इफेक्ट रिलेशनशिप नहीं आती. इस तरह ये आरसीटी में भी जो है कॉमन होती है एक्सपोजर एंड आउटकम, बट इट इज एक्चुअली द रिस्पॉन्सिबिलिटी ऑफ द researchers to maintain it very clearly and very explicitly clear about what is the exposure and what is the outcome. The next is the performance bias. Now, what we mean by the performance bias, the last one in the trial implementation, the bias during the trial. The performance bias they occur when difference in the care provided, aside from the intervention being studies, affect the outcome of the study. This type of bias that typically arise when participants or researchers are aware of the treatment allocation that leading to unequal treatment or effort between groups. One of the best solution to control such performance bias is the not only the randomization but also the blending, and specifically the blending of the assessor, the blending of the patient. That is very important. So, what are the causes of the performance bias? So, some causes of the performance bias they can be knowledge of the group allocation, unequal co-intervention, the placebo, the expectation effect. Uh, when there is the researcher or the assessor or the therapist, if they have the knowledge of the group allocation, so the participant and the researchers which are known to such a location, they may behave differently because they know which group is the control, which group is the treatment, and in which group they are. They are in the control group, they are in the experimental or treatment group. For example, a physical therapist might provide more encouragement to participants in the treatment group, and at the same time, they are giving less focus, less concentration to the participant which are in the control group. So, such uh, uh, no knowledge about the control and experimental may lead towards the performance bias. The next reason is sometimes participants in one group receive additional intervention or support that are not part of the study protocol. So there may be the violation of that protocol. They may lead towards the performance bias. For example, patient in a new drug trial receive more frequent follow-up than the control group, which is again considered and called as the performance bias. Similarly, the effects of placebo are that can also be affect the cause lead towards the performance bias. The participants are the that researchers' expectation about the treatment can influence the outcome. For example, participants in the treatment group may believe they will improve and thus report better outcome. And that is the reason why we are again and again saying that the assessor, the therapist, and the patient they all should be blind during the trial, because when they all of them they are blind to the location, so they will be unknown to the which group is treatment, which is the control group, and the patient who they are treating, who they are assessing, is belonging to either control group or the experimental group. So they will have no such effect on their performance. So there will be the control of such bias. So these all bias, the interviewer bias, the chronology bias, the recall bias, the transfer bias, misclassification of exposure and outcome, and the performance bias, they all are the bias which occur during the trial, and they are occur during the implementation of the trial. There may be some other problem like the citation bias or the confounding. We have already discussed the confounding in detail. Citation bias, they occur during the publication, mean we are missing some citation to the statement and we consider them the statement that they are our own statement. They are when we are consider someone idea, someone results to the results of someone else, there may be some problems in the citation, or we are just citing those studies which are in favor of our study and we are not citing those who are not in favor of our results. Similarly, we are only reporting the old studies, we are not reporting the new studies, are just we are reporting only those studies, either they are old or new, they which are just in the favor of our results. So that is again called as the citation bias. So that was all about the bias and their types. There are different types of RCTs, or we can say that the clinical trial, they may be on the base of the intervention, they may be on the basis of how the participant are exposed to the intervention, or we can say they can be the types of the RCTs, they can be on the number of participants which are involved in the research. Uh, Similarly, the RCTs can be divided on how the intervention was assessed, or the preference types. So different types of RCTs we will discuss each of them specifically. We will discuss here what we mean by the explanatory and the pragmatic or the practical trial. So the pragmatic trial, they are designed to evaluate the effectiveness of intervention in the real world routine practice setting, unlike the explanatory trial, which assesses the efficacy of intervention under tightly controlled condition in the laboratory. So the pragmatic trial, they mostly focus on how the treatment performs when implemented in everyday clinical practice, while in the explanatory trial, usually uh, There is checking about the dose, how many dose will be enough, how much dose is bearable, what are the side effects, if it is safe for the use in the complete dose and the participant in during the routine daily life, what is the mechanism of the drug, what is the pharmacokinetics of the drug, and all other pharmaceutical parameters they are checked during the explanatory trial. While how they work and the real patient, if we are doing the randomized control trial while recruiting the patient and treating the patient in the hospital, in the clinic, in the real world, these trial will be called as the pragmatic trial. We will discuss each of them in the upcoming slides. So first of all, we will talk about the clinical trial phases. These the drug trial, basically these are most commonly used for the drug trials. These trials, they can be of different phase. They can be the preclinical, the phase 0, the phase 1, phase 2, the phase 3, or the phase 4. In the preclinical, the primary goal of such trial, that is the testing of drug in non-human subject, just to gather efficacy, toxicity, or the pharmacokinetic information. Basically, these studies they are conducted in the lab and just to find out how much side effects they have and what is there any efficacy of this drug or not, and what are the other pharmacokinetic information, different parameters of this drug. The dose is not still decided, so this is unrestricted. The scientific researchers they will monitor the patient, and there is no such participant required for such trial because these studies they are in vitro are in vivo only, so that is the number of participants is not applicable to this preclinical trial. In the phase 0 of the pharmacokinetics, particularly the oral bioavailability and the half-life of the drug is being checked and assessed during this trial. The dose which is provided to the patient, that is very small and that is less than the actual dose, because एक्चुअली यहाँ यहाँ पे हम इफेक्टिवनेस नहीं देखते, हम सिर्फ देखते हैं कि इसकी बायोअवेलेबिलिटी, इसकी जो हाफ लाइफ है या इसकी मैकेनिज्म इसकी क्या है. So for that we just need a very small dose because we are not checking when assessing the effectiveness of this drug. The clinical researchers will monitor the patient and mostly up to the 10 people. There might be 7, 8, 9 or less than that, but up to the maximum 10 people, usually the volunteer people, they are just enrolled and recruited for this phase. Somehow, if we have such information in the preclinical trial, so this trial sometimes they may be skipped and directly from the preclinical. The researchers they shift, they transfer their study to the phase 1. The the primary goal of the phase 1 is just the testing of the drug on the healthy volunteers for the dose ranging, that how much therapeutic dose would be enough to treat such a disease and how much therapeutic dose will be bearable by the patient, by the person, by the human without any serious side effects. So often this dose is subtherapeutic but with ascending doses. It means there is gradual increase in the dose and each dose is monitored, and in the dose in which there are some serious complications or serious side effects, so the dose below that is considered as safe and then being reported. The clinical researchers will report the patient, usually 20. The lower limit of the participant in this study is 20 and the upper limit is 100. Between them, they may be in or voluntarily recruited for this trial, and all of them they will be healthy. And the success rate is approximately 70%, which means If we are in the some drug is being moved toward the phase one, so it means that we are 70% sure that we will achieve the goal of that goal of having the therapeutic dose or the dose range for that drug. And at the end of this study, at the phase one, the researchers they reported that either this drug that is safe for the humans, it is it can be used further, or it is unused, or there are some serious side effects. So there is notes reported that this drug shouldn't be used for such diseases in the patient because there are serious side effects. In the phase two, the dose is already been decided, the pharmacokinetics is already been, and the researcher is being informed. The researcher has been known to the toxicity and efficacy of the drug. So, what is the primary goal of the phase two? So, actually, there is testing of the drug on patient to assess the efficacy and side effects. So, this is actually the first time when the drug will be used on the real patient. Therefore, the dose which will be given to the participant will be therapeutic dose. The clinical researchers will monitor the patient, and the number of participants will be gradually increased. As there were 10 people in the phase 0, there were 20 to 100 in the phase 1. Now, in the phase 2, the number of participants will be from 100 to 300 patients. Before that, there were healthy volunteers, but in this phase 2, they will be patients with specific disease. And there is approximately 33% chances of the success rate. So in the end of the phase two, the researchers reported that if they determined that whether the drug can have any efficacy at this point, the drug is not presumed to have the therapeutic effect whatsoever, and after that, the drug is being either stopped or either checked in on another large group of population. In the phase three, the studies when there is success in the phase two, and there is no such serious side effect reported. There is no side effects. There is no, and there is also good effectiveness, excellent effectiveness, high effectiveness of their drug. So then the drug is being assessed on a very large number of people, which range from 300 to 3,000 patients with a specific disease. The primary goal of the phase 3 is the testing of the drug on patient to assess efficacy, effectiveness, and safety. Just to reconfirm what we have achieved in the phase 2, the dose is therapeutic dose, and the clinical researchers and an experienced physician they monitor the patient, and the success rate is somehow reduced because the number of people now they are in thousands, so that's why there may be less success than the phase two. In the phase four, when the drug is being considered safe and is ready to be used for the general population, that may be available in the market as well. So in the phase four, the primary goal for such trial is the post-marketing surveillance, watching the drug use in public, that is in the market, in the shop, in the medical shops, in the pharmacy shops, in the pharmaceutical stores. And if there is some serious side effect reported by a number of people in the public, then this drug is being banned and being stopped from selling it by the shops, and is being collected from the shops. And if there is no such reporting of bias, reporting of side effects from the public, so that is the now the practice and the use of such drugs, such medicines, then continued by the public and by the shop and physician. So in this case, the researchers is not available, as now the drug is available in the market. So the patient they will be monitored by the personal physician, and there is no such estimate on number of participants and how much they will be in this phase. Anyone who is seeking the treatment from their physician may be the part of this study, and success rate this is may not be reported as we don't have the complete data of all the people who have been received that treatment. And in this phase also, this is being checked by the researcher or by the team or by the pharmaceutical company for its long-term effects of their drug. So these are the different phases of the trial which are being used for the drug trial, the preclinical, the phase 0, the phase 1, the phase 2, phase 3, and the phase 4. The explanatory and the pragmatic trial, we have already discussed them previously that pragmatic trial, they are the trial which are conducted on the patient in the real life in the hospital, while in the explanatory trial, they are basically conducted in the lab just to assist, just to determine the dose range, just to determine the safety and the side effects, and the mechanism, the pharmacokinetics, etc. So that will be known as the explanatory. The explanatory trial designed to see if a new treatment drug works, very strict including criteria to exclude coexisting disease, generally have a placebo as control, and usually they are checked under a very strict control environment. On the other hand, pragmatic trial designed to determine if the intervention worked but also to describe any consequences of its use under circumstances that may make the clinical practice wider, including a criteria to ensure the population represents the normal group of patients often compared with the current treatment or the control. Now, the different types of design of the RCT. So one of them is the simple RCT design, also known as the parallel design. This is when suppose there are 20 participants and they are allocated randomly into two groups, into the group A and the group B. The group A is the experimental or the treatment group, and the group B is the control group, or it is we can say that is the treatment two. So those participants who are allocated to the treatment A, they will receive the treatment A, and those who are allocated to the treatment 2 are the B group, so they will receive the treatment 2 or the control group protocol. Till the end, they will be both followed by equal number of session, equal number of days, and then they will be compared. So that simply it is known as the parallel design. The next is the crossover design. Suppose sample is recruited, then the sample is being randomly allocated to two groups, the group A and the group B. The group A participant which were allocated randomly to the group A, they will receive the treatment 1, and the participant who are allocated to the treatment, the group B, they will receive the treatment 2. After some time, the participant in the group A, they will be shifted to the group B, and the participant in the group B, they will be shifted to the group A, and they will the participant who have received the treatment 1, now they will receive the treatment 2, and the participant who have been received the treatment 2, now they will receive the treatment 1. There is a specific duration between the exchanging these treatment with each other, and that time period is known as the washout period. This washout period is very necessary just to eliminate any effects of the treatment 1 or 2, because if there is still some effects of that treatment exist, so then the effects of the post treatment, the 2 and 1 in this case, they will be then mixed with the effects of the previous two treatment. Hence, there may be no such clear conclusions. For that, there should be enough time to wash out or to eliminate the effect of the previous treatment. So hence there is exchange of the both treatments. So that's why this is known as the crossover design, also known as the repeated measure design. Why the repeated Meyer design? Because here the number of assessment there will be at least three times. Mean they will be baseline assessment, they will be assessment at the end of the trial, and there may be then assessment and the end of the another two treatment. So that's why when we have three or more than three time data collection from the same participant in any clinical trial, that is known as the repeated Meyer design. And if we are using the statistical test for them and the data is normal, so the repeated Meyer ANOVA will be used for them in the within the group analysis. That what is the difference between the baseline similarity, the second assessment, and the third assessment. So these all will be. So this is equally the crossover design or the repeated Meyer design. The next design is known as the pectoral design RCT. In such RCT randomized clinical trial, one group out of the three experimental group that is equally the subset or the combination of the other two or three treatment. Suppose the group A they are receiving the treatment 1, the group B they will receive the treatment 2, and the group C they will receive both the treatment 1 and treatment 2, and the three of them they will be compared with another control. So this is actually known as the uh, RCT. But the thing which is important is the in these four groups, the participant of all the recruited, the selected participant for this trial, they will be randomly located to all these four groups. Suppose we have 60 participants, 15 participants will be located to each group, 15 to A and 15 to B, 15 to C, and 15 to D. We have discussed them in detail in the previous slide. So just remember this thing. And the phase one, this is the first human exposure. Phase two, this is large scale human exposure. And phase three, clinical trial, which is up and randomized. And in the phase four, this is the post, that is the surveillance of that drug in the market. Another is the sequential trial. Sequential trial are those to design in which the number of participants they are not specified at the start of the trial, and the recruitment continues until a clear benefit of one of the intervention is observed or not important differences between the intervention can be detected. The next is the comprehensive cohort design. This is a very special case of the randomized control trial in which the eligible participant, when they are asked for the randomization, so some of them they agree to be randomized to each to either to group A or group B, but some of them they deny to be randomized to either the group A or group B, and we have a very shortage of the participant, or we are just treating a very rare disease. The patient of such disease they are very rare and difficult to find. So we agree to the conditions what the patient have. So those who agree to the randomization, they sign the consent for the randomization. They are randomly allocated to the treatment A or treatment B. On the other hand, or denied to be randomized, so they are then asked which intervention do they prefer. So those who prefer the A, they get the A, and who prefer the group B, they get the B. And hence, in a single trial, both the randomized and the non-randomized control trial, they are conducted and executed, and then they both groups they are compared with the control. Ideal pathways where a high proportion of individuals are likely to refuse the intervention, and the disadvantage of comprehensive core design is that difference observed in outcome may be due to the difference across the randomized and non-randomized group at baseline. Single blind trial, already we have discussed them in detail, what we mean by the single blind trial, the double blind trial, and the triple blind trial, and also there is quadruple blind trial. A single blind trial, either the researcher or the patient does not know about the detail of the treatment, or what is the experimental treatment, what is the control treatment, or who are in the control group, who are in the experimental group. So the patient or the researcher, some one is the primary researcher, the main investigator, the chief investigator, and the other investigators, which are the group of the researcher, they may also have no knowledge about the treatment allocation. If the patient is blinded, this may reduce the likelihood of a placebo effect. If the research is blinded, this may reduce the likelihood of the ascertainment found out for the certain bias. Double blind trial. In a double blind trial, one researchers allocate a series of numbers to a treatment group and control group. The second researcher is told the numbers but not what they have been relocated to, so the patient is not told which information they have received, reduced likelihood or placebo effect or ascertainment bias. In double blind trial, usually the patient as well as the assessor who is recruited for the assessment both before the treatment, after the treatment assessment of the patient, both of them they are blind is known as the double blind trial. There is triple blind trial in which the assessor, the therapist, and the blind and the patient, all of three of them, they are blind to the location. Quadruple mean when the patient, when the assessor, when the therapist, and the one who is involved in the statistical analysis, they all are blind regarding the allocation of regarding the treatment group, who is the experimental, who is what whose treatment is the control. So that will be called as the quadruple. But in case if there is no blind, is mean no one is blind to the relocation of the treatment group, so that will be called as the open trial study or open label trial. Biases in the RCTs, we have discussed in detail about the selection bias. The ascertainment bias, assessor or participant knowledge of intervention blending can reduce potential for the ascertainment bias. What are the advantages of the RCT? That allows rigorous, extremely thorough, accurate evaluation of a single variable in a different patient group, potentially eradicates bias by comparing two or more identical groups, allows for the meta-analysis. What do you mean by the meta-analysis? The statistical procedure used on the results of different studies to make a single report, make a single result combined from the number of RCTs which have been, and all of them they are focused on a single problem, a single treatment is being used, and the patient are similar, so that is known as the meta-analysis. What are the limitations of the RCTs? One is the external validity. External validity is the confidence you can have in generalizing your results or finding across the people, situation, and time not included in your study. So it may be not necessary that the results which have been reported, published in the trial, that exactly they should be the similar result in other settings, the other patient, because there may be variance in the patient's gender, in the patient age, in the patient severity of the disease, as well as in the procedure, how the treatment is being provided. So it means there may be variation in some sort of. So that's why the biggest limitation of the RCT is the external validity. That sometimes there may be no such similar result in the patient who are not included in the study in the other areas in the other setting, like the reports of the published study. A study may have high external validity with regard to people. For example, a random sample of 1100 likely voters, but poor external validity with regard to time. For example, the sample was collected in 1960. Patient recruitment, when there is a risk of being allocated to an undesired treatment option, patient compliance may be low, and that's why randomization is one of the procedure uh, which can help in the controlling of such uh, problem, such risk. Attrition mean in the RCTs they are based on clear statistical laws of probability. Researchers undertakes accurate calculation of the required sample size to demonstrate a difference between group. What happens when you have patient drop out or missing data? ट्यूशन बाइस उसको कहते हैं जब एक स्पेसिफिक ग्रुप से बहुत मतलब सिग्निफिकेंटली लोग जो हैं ड्रॉप आउट हो जाते हैं उस स्टडी को छोड़ के चले जाते हैं so that will be called as the attrition bias. So this attrition bias is also one of the limitation problem in the RCT. Other limitation, other problem is the researches you can face during the conduction of RCT. They can be very expensive and as well as very time consuming. Often have two patient or too short period of that follow period and often imperfect randomization and often not all eligible patients are randomized. So that was all about the what is RCT, what are the types of RCT, what is bias, what is the types of bias, what are the advantages and limitation of the randomized control trial. That was all about. Thank you so much. Have a nice day.